Our next speaker is Sonia Batten, and she'll be talking about the Continuous Plankton Recorder Survey on a transect through the Aleutians.
Thank you. So when Matt made me aware of this forum, I thought it would be a really great opportunity to come and talk about this project to a new audience. And it also gave me a nudge to actually look at the data we've been collecting in this region, because so far I've been focusing on other areas and hadn't spent much time looking at this. So it was a good opportunity for me to do that. So I'm going to be talking about plankton, and in case anyone in the room doesn't think that plankton are the most important things out there, let me tell you they are. So they're the basis of every marine food chain, pretty much. They support directly or indirectly all of the resources that we value. Because they have short life cycles, and because they have very limited ability to move, and because they're not commercially fished, or almost exclusively not in this area anyway, they react very quickly to changes in their environment in a relatively unambiguous way. So they're a good indicator of what's going on in the environment. So plankton are important. And what I'm going to be talking about here is the Continuous Plankton Recorder Survey. This uses commercial ships. It's now the Matson Kodiak up in Anchorage. These use commercial ships to tow a plankton sampler behind them along their regular route. The plankton sampler filters the plankton out of the water as the ship goes onto a long length of net. When we get it back from the ship, we cut that net, use the ship's log information to cut the mesh into individual 10 nautical mile samples, which basically reconstruct the ship's voyage. And using the ship's log information, we can give each sample a position, a time, and date of where we got it from, reasonably accurately, at least on the scale that I'm talking about here. And then we take those samples, and then people have to sit down at a microscope and look at all of the, I know, that's the tedious, time-consuming bit, and identify the plankton that we see. So we end up with a record of different taxa and their abundance from different parts of the ocean. And then over there on the bottom left, I've got a close-up of the mesh. So the filtering size the mesh size is about 270 microns, about 1/4 of a millimeter. But I always make the point that the mesh is actually more mesh than whole. It's about 40% porous. So we do catch things that are smaller than 270 microns too. Okay. So over on the top right here, that's the entire North Pacific CPR survey positions. And then I've zoomed in onto the area around the Aleutians. And what I've got there, the yellow dots mark the center of each 10-mile sample that we have analyzed. Because it's expensive to have people sitting down and doing the samples, we analyze every 4th sample from the transects, and then we archive them all. So they're all available for reanalysis. But so what's on the bottom there is just the samples that I have data for. And as you can see, the ship pretty much always goes through Unimak Pass on the way in. Where it exits depends on a couple of things. Sometimes they're avoiding storms. Sometimes it just depends on which port in Asia the ship is heading to. So it's a bit more variable in the west. And then just to go through what the kind of data are, in that Aleutian Island region, we've got about 90+ phytoplankton taxa. The CPR samples the larger diatoms and hard-shell dinoflagellates pretty well. Pretty good record there. Smaller and single cells are not so well sampled. And the group, the naked flagellates, we just don't see at all. The preservative just doesn't keep them in any good condition. And then we've got about 130+ zooplankton taxa. And the CPR samples crustacean plankton really well, so copepods and euphausids are well sampled, particularly in the size range of 200 microns up to a centimeter or so. Organisms smaller than this or larger than this may not be well sampled. And if they're particularly fragile, we may not be able to identify them with any certainty. So things like gelatinous plankton, it looks like somebody sneezed. That's pretty much as far as you can go. And then just a few additional caveats. Sampling takes place in spring, summer, and fall. We do 3 transects a year. The ship takes about 2 to 3 months to do a complete voyage from the west coast over to Asia and back again. So we do spring, summer, and fall. Obviously, we have no control over where the ship goes and when it leaves. We get what we get. And we consider this a surface 0 to 15 meter mixed sample. We tow horizontally. We put the machine at around about 7 meters depth, but these are big, fast-moving ships, and it's quite turbulent behind them. So we think of it as a surface sample of plankton. And I should say that we first did this in 2000. North Pacific Research Board have been big supporters of this program, and they've contributed some funding right since the early days. So we now have 15 years of data, which is... There was a lot of talk this morning about baseline data and needing baseline data, and as the NPRB are considering potentially focusing on the Aleutian Islands, it's very useful to have a good long time series of baseline data that you can make assessments about changes against. So for the rest of my talk, I'm basically going to just give some examples of some of the data analysis I've done, just to it's certainly not exhaustive, it's just to whet your appetite, and I found it interesting to do too. So what I've done for all these data is I've subdivided them into 3 regions: the east, which is pretty much the shelf and slope region through Unimac; the central region, which is pretty much all deep water; and then the west, which is a bit more variable in position, but mostly deep water. And so for the next slides that you see, that those regions will always be referred to as the same. So the first thing I did was look at the seasonal cycle of the phytoplankton. I took the diatoms; they're the most abundant group that we see. And you've got 3 seasonal plots here. So this is 15 years of data amalgamated. And not too many surprises. So for the eastern region, you see what we typically find in a shelf system: a fairly even spring and autumn peak in abundance. In the western region, we have really only a spring peak in abundance, and then it sort of tails off through the summer. And one thing to note is that there's not really a big difference between the 3 regions. The numbers are roughly similar. And we only sampled in the wintertime once, but you get very little phytoplankton around in the wintertime. And then if we have a quick look at some zooplankton data, so these pie charts are the broad community composition, so representing by abundance the different types of zooplankton. I've got the 3 regions in left to right, and then spring, summer, and fall averages. So copepods dominate the community. These are the ones, the light blue ones are the small copepods, and the dark blue ones are the large ones. And I don't know how well you can see it, but there's a picture in the bottom right of typical small and large copepods. So the biggest ones there are roughly 4 to 5 millimeters long. And in those you can see a little sac that has fat in it, and that's why these things are very tasty food sources, because they accumulate fat through the spring and summer. And so they're pretty good. So they're not as numerically dominant, but because they're many times bigger than the small ones, they make up a lot of the biomass, particularly in the spring and summer. And then there are some regional differences. So although copepods dominate throughout, you can see that red splodge over in the western region, which is the pteropods. Those are those small pelagic snails, and they are more important, seem to be, in the western region. And then some of the other groups that show up, the gray ones are, as I've called it, other mesozooplankton. Those are typically the larval stages of some of the crab and shellfish that are only maybe in the surface waters for a small amount of time. So their numbers can fluctuate quite a lot. So we do see some regional and seasonal variability in what makes up the zooplankton. So then I thought I'd take a look at the time series. So we have more data for the summer samplings because there are fewer storms, so everything works better. So I have data for most years from 2000 to 2014. And this is the total diatom abundance for the summer transect for each of those regions. And the first thing I noticed is that I've got an alternating pattern of high and low abundance for each region all the way through, at least until 2012. And I don't usually see this. Usually I see trends that correspond to ocean temperatures, so a period of warmth when things might be high or low. So having this interannual fluctuation was really unusual. And I wondered why. What would be causing this? And pretty much the only thing I could think of that has a 2-year repeating cycle like this is pink salmon abundance. And there certainly have been work by Japanese scientists looking at pink salmon and their relationship with zooplankton in the northern Pacific, just south of this region. So I had a look at that, and we do see a pretty strong positive relationship between diatom abundance and pink salmon abundance. So it's much stronger in the eastern region, very significant, and although it's there, it's not significant in the west, but it's also there in the central region. So I thought that was pretty cool. And then I wanted to have a look at the community composition. So all you need to take from these diagrams is that the little circles have a number in it which represents the year. The more similar 2 years are in their community composition, the closer together that they plot, and the more different they are, the further away they plot. And what you can see in the western region over there on the left is that there's no real pattern, there's no real clusters, but there's kind of a change through time. So the early years tend to be more at the bottom of the plot, and the later years tend to be at the top. So there's a slow, gradual shift in community composition over time in the west. In the central region, most of the odd years cluster together up in that top right in that oval I've put, with the exception of 2003. And then the other years are kind of spread around. But in the eastern region, I get a clear shift between odd years and even years. So everything to the left of that line is an even year sample, and everything to the right of that line is an odd year sample. So potentially, perhaps, there is an influence of the salmon on the types of phytoplankton that are out there too. So then I looked at the zooplankton, and I picked the large copepods because they're principally what is providing a food source for fish, as well as other organisms out there. And I saw the same thing in the central and eastern region, an alternating pattern, this time lower in odd years and higher in even years. And we don't see this pattern in any of the other zooplankton groups I've looked at, but in terms of biomass, these things are the most important in the spring and summer, so I focused on these. And again, if we compare that to pink salmon abundance data, then in those years with high pink salmon, we find low abundance of copepods. And this is very strong in the east, also significant in the center. So we'll put that together for a potential linkage. Then I've just chosen the eastern region here. So what I've got here are bars showing the average odd year abundance and even year abundance for the pink salmon. And you can see there's quite a difference between odd and even years. And then for the large copepods and for the diatoms. So what it looks like is happening that in odd years, there are lots of pink salmon. They eat lots of large copepods, reduce the grazing pressure, and allow the diatoms to be more abundant. And the reverse is true in the even years when there are a few pink salmon. They don't eat so many large copepods. That means the large copepods can graze on the diatoms, and we have fewer diatoms. So this was pretty cool, because although other people had found this for a few years in the 1990s, I have never seen a top-down impact on my data. Usually I don't see any influence of fish. I see much more forcing by the environment on what the plankton are doing. So this was quite exciting. And the second thing that was exciting is like, well, what happened in 2012 or after 2012? Because the last 2 years I don't see this pattern that I would expect. Apparently ADF&G recorded their highest ever pink salmon abundance in 2013, so it's not that pink salmon weren't abundant then. So we should have had high abundances of pink salmon eating the copepods, but there was still plenty. In fact, for some of the regions, the highest overall summer abundance was in 2013. Diatoms were also still high, so even though there were lots of large copepods, they weren't being grazed. 2014 was more typical, but there were still more diatoms. And then on the bottom here, I've got, I know that it's not great for people over the other side of the room, but I've got, for large copepods and diatoms, the average odd and even year patterns, and then 2013 and 2014. And they don't fit. The other thing that doesn't fit was when I looked at the microzooplankton. Now, the plankton recorder only catches a small proportion of them. These are the very small ciliates that have a hard shell. So I've got a picture of a tintinnid there, and foraminifera, another one. And they were really high in 2013 and 2014, much higher than we've seen before. And one other group, just to talk about briefly, I said that gelatinous plankton we can't really identify. So we can't count them, but we do see the stinging cells of jellyfish in the samples. And so we've always recorded presence or absence. And if you assume that there's some relationship between the number of samples that you see stinging cells on and the abundance of jellyfish in the environment, then this may be a record of jellyfish abundance. So over here I've got the data for, this is for the whole year for the 3 regions. And the warm year of 2005 shows up as a peak. That's pretty common, and 2014 also. But I thought it's worth making the point that in all 3 regions, jellyfish were potentially the highest abundance we've seen in 2014. So just to try and summarize then, so what I've shown are plankton data from almost 900 analyzed samples collected near the Aleutian Islands from 2000 to 2014. And I've split them into 3 subregions, and you can argue whether there could be better definitions of those regions. But there's an archive of over 3,500 samples now, which could be reanalyzed. And there's also other cool things you can do with the samples. You can do some DNA work. You can look for specific groups if you want to focus on those. So the archive is there. We can see that seasonal cycles for phytoplankton, and I could show them for zooplankton too, and community composition can vary regionally. And then we have this alternating pattern of high-low years of abundance, which seems to fit with control by pink salmon. Now, if the pink salmon are consuming a lot of the copepods, that may mean that there's fewer of them for other things. But I think one thing that I'd like to follow up on is that after 2012, so 2013, 2014, we get something unusual happening. We see unusually high abundances of things that we hadn't expected. And this suggests that something in the ecosystem has changed. And I don't know what that is yet, but I'm open to ideas. So I just want to finish thanking you for your attention. The data are freely available. I'm always happy for collaborators, people using that. So if you want any further information on the data, that's my email address. And then I'd just like to acknowledge the officers and crews of the shipping companies. This is a pristine region, and there's a lot of ships that use it, and I know there are a lot of implications, potentially bad implications, but having the shipping communities at least helping us is a small positive step, at least. They're providing some environmental data, and they're all very pleased to be involved. The environment does matter to them, so we've always got really good support from shipping companies. And then my local personnel in Anchorage and in British Columbia, where I'm based, who do a lot of the work, and all of the people who spend numerous hours at the microscope, myself included, to get the data. And then last, of course, the funding contributors, North Pacific Research Board, Fisheries and Oceans Canada, the Exxon Valdez Oilspill Trustee Council, and the North Pacific Marine Science Community. So thank you.